So in this video, we're going to talk about enzymes and how they work. Enzymes are proteins that speed up chemical reactions. So they're called catalysts, and a catalyst is a protein that speeds up a chemical reaction, but is not used up in that reaction. So enzymes are catalysts, and their major function is speeding up a chemical reaction. They do this by lowering the amount of energy needed. So basically, an enzymatic reaction does not require near as much energy as a non-enzymatic reaction. And so what is that energy that we need for reactions to occur? It's called activation energy. So this is like the little symbol for the activation energy or energy of activation. This is the amount of energy that has to be added for a reaction to occur. And so with enzymes, we decrease the amount of activation energy needed. And so when this happens, enzymes then can so they have to bind with molecules, and when they bind, they're either going to stress the chemical bonds, or they're going to break those bonds. Is that weird? I swear that's so weird to me. Break those bonds. And so the reaction would occur without the enzyme, but with an enzyme, it's going to increase that reaction rate. So all reactions would eventually occur, but enzymatic reactions occur much quicker. And so enzymatic reactions can either build up or break down molecules. And so let's look at this. So here we have reactants. Reactants here have a higher energy. And since it's a higher energy here, and that is not a Y, is it? No. E-N-E-R-G-Y. And this is lower energy. We're looking at a reaction that is releasing energy. So if you remember, that means it's a catabolic reaction. And since we have something that's higher in energy in the beginning, then it's also an exergonic reaction because we're releasing energy. And if you remember when I was talking about it, I said catabolic and exergonic reactions always are tied together. So without an enzyme, this reaction would occur, but it takes a lot more energy. So this is the amount of energy that is required for this reaction to occur. You can see that blue box is a lot more energy there. This yellow box is a very small amount of energy. So with an enzyme, you're going to lower the amount of energy needed, and so you're going to increase that reaction rate. So enzymes have a region known as an active site. And the active site is where the substrate binds. And so here it says active site. Here's the substrate. If you look at the substrate, it fits in the active site. So enzymes are specific to their substrate. And so one enzyme typically will only catalyze reactions with one substrate. But it can catalyze multiple reactions because enzymes don't get used up. So in this case, the substrate binds to the enzyme's active site, and we're going to complex together, so the enzyme and substrate complex form, and that enzyme is going to stress, or is going to break the bonds between these two molecules. So you can see here's the boxy molecule, and here is the like, I don't know, unique looking molecule. Looks like a house to me, but and here you can see them starting to separate. And once the enzyme reaction occurs, then the products are released. So, and the enzyme is free to be used again. So this is a degradation reaction. So we have the substrate is broken into two smaller products, or into smaller products. So this is also a catabolic reaction. And it is an exergonic reaction. Okay. But enzymes can also build stuff up. So here, again, we have the active site. Here's the active site. Here we have two substrates, the same two substrates actually. And these two substrates bind to the enzyme. And once they bind in that active site, the enzyme and substrate complex together, and they stress the bonds. They put energy into the bonds, forming that product. So we took in energy. This is an endergonic reaction. And it is an anabolic reaction. So enzymatic reactions pretty much occur in every single metabolic reaction that occurs in a cell. So you can just assume at this point that all metabolic reactions occur with the help of enzymes. So without enzymes, reactions will still occur, but they will occur at such a slow rate that life could not exist. Hello? Oh, nope, that's not what I wanted. Sorry, I went backwards. All right, so enzymes have what we call an induced fit model. We used to think or assume enzymes fit like a lock and a key. So you have a lock that only fits with one key. Enzymes only work with one substrate, but we now know that enzymes are not static. They're not immobile. They can change in shape slightly. And so now we know that the induced fit model is the best model for how enzymes work. When a substrate binds to the active site, the substrate and the active site typically don't match perfectly, but as soon as the substrate starts to bind, the enzyme changes its shape slightly, kind of giving that substrate a little hug. That's kind of how I look at it or think of it. And that change in shape actually activates the reaction that's going to occur. Oh, good Lord. Okay, and so here you have an enzyme active site. And so this is a much better looking active site than that, you know, how I normally draw them or how the other one was drawn. It had like a static spot. This is more like what an enzyme active site looks like. And then the substrate binds and the enzyme kind of conforms to the substrate, either building or breaking those bonds. And that's the induced fit model, okay? I'm trying to change to the next slide. There we go. So the last thing I'll talk about is how we name enzymes. So every single molecule typically requires their own specific enzyme, and enzymes are typically named for the substrate they work with. And so here are some examples. Lipids are broken down by lipase, urea by urease. And so if you notice, the enzyme names all end in ase. The suffix A-S-E, is typically what we see in the name of the enzyme. That's not always true. An example of an enzyme that doesn't have that is pepsin, P-E-P-S-I-N, works on proteins. It's an enzyme and it doesn't have the ACE, but almost all enzymes do have that suffix ACE, so it's easy to identify enzymes. I'm going to stop here and we'll continue on in enzymes in our next video, okay? Bye.